All right. Welcome, everyone. I am recording. This is Peter Smallage. I'm the New York State Extension Forester. I run the Forest Connect program, which includes this monthly webinar series. And it had occurred to me that we had maybe never or not frequently had any kind of discussion about trees that get sick. So the forest pathology and tree pathology side of the things that we do. And so I started casting. Cornell used to have a forest pathologist a long time ago, not now. So I started casting about, and the name that came up as somebody that'd be great was Dr. Nick Brazee. And so I reached out. He was happy to help. And today we are going to learn about an introduction to forest pathology. And it's part of the first of a three-part series on forest health, if you saw the email notice. So I'm going to turn off my screen and microphone, and I'm going to turn it over to Nick. And Nick will give a more thorough introduction and then talk to us of himself and then talk to us about what makes trees sick. So, Nick, thank you so much. I'm really looking forward to this presentation. I appreciate your
Sure.
time.
Thank you, Peter, and welcome everyone. Good afternoon. Yes, as Peter said, my name is Nick Brazee. I'm an extension plant pathologist here at UMass, and I primarily run the diagnostic lab. So I get anywhere from 800 to 1,000 samples a year, primarily from arborists and landscapers, also homeowners. So try to figure out what is happening with trees and shrubs and provide some corrective action. My background is in forestry and forest pathology, but through my work here at UMass, I primarily worked with urban trees and suburban trees. But for today's lecture, I'll try to keep it forest-based with the examples. There may be some urban trees that make their way in, but it'll still provide a nice background, I hope, for the subsequent lectures over the next 2 months on beech leaf disease and also eastern white pine health issues. So I'm going to go over some of the basics of forest pathology, some of the core concepts of tree pathology, and provide some examples to kind of illustrate those. So we will dive right in. Let's see, I'm going to turn off my video as well. All right, so let's dive right into this. We're talking about tree disease. What is disease? What is disease specifically? If we have a technical definition, and this comes from George Agrios. He wrote the book of plant pathology, used in a lot of plant pathology courses. He was a professor at the University of Florida. Prior to that, he was a professor right here at UMass when we had a Department of Plant Pathology many decades ago. I'd like to think that George didn't leave UMass because he didn't like UMass. I'd like to think that it was, maybe after a winter like we just had, and he said, I'm getting the hell out of here and going to Florida so I don't have to shovel snow. Now, from Agrios, disease is any malfunctioning of host cells and tissues that results from a continuous irritation by a pathogenic agent. That would be a biotic agent. We'll go over what that means specifically, or an environmental factor, and leads to the development of symptoms. And I will talk about symptoms as well. Okay, so a pathogenic agent, which are biotic agents, or environmental factor. And just so we are all on the same page, biotic are living organisms. That would be fungi, oomycetes. If you're not familiar with oomycetes, they are fungal-like organisms. And maybe you've heard of Phytophthora or Pythium. Those are oomycetes. Bacteria, viruses, nematodes, and parasitic plants are abiotic. Disease agents would be anything non-living, drought, pollution, flooding, wind, and fire. And so I think it's really interesting to think of these abiotic agents as causing disease. And what we have here, so this map on the left, this is from the Northeast Regional Climate Center based out of Cornell, and it's showing percent of normal precipitation from September through November 24. If you remember, the fall of 24 was historically dry. Now, the way you interpret these maps, when there's a lot of green, and the darker the green, that means we're above average. And when there are shades of orange and red, that means we're way below average. So it was very dry across all of the Northeast and some areas in particular, including most of Massachusetts, but big swaths of Maine and eastern Pennsylvania were very, very dry. And so to think about drought stress as causing disease, I mean, typically we think of drought as just a stress and not a disease agent itself. But if we are to go by the definition that Agrios established, it fits the criteria as being responsible for disease. So I always find it interesting to think of those non-living, those abiotic agents as being responsible for disease. Now, drought is probably the most important predisposing stress when it comes to disease. And I'm going to talk about some specific examples there. But a lot of disease is driven by weather patterns. And we will come back to that time and time again. With insects, they're a bit more independent. They have, there's the growing degree days when they emerge, and if it's wet or it's dry, that doesn't influence them as much. But a lot of disease outbreaks are really reliant on the environmental conditions in that particular area. Okay, so we've established what disease is. We have our biotic agents, those are the living. We have abiotic, they're non-living. Armillaria is one of our biotic disease agents. I would hope that nearly all of you are familiar with armillaria. It's one of the most, if not the most important decay pathogen here in the Northeast. Armillaria is fascinating because it lives as both a parasite, it can parasitize a living host, and it also has this ability to live as a saprophyte. So after it's killed a tree, it can live for years, if not decades, on the stump or within the roots as they decay and break down. Armillaria also has the ability to produce this melanized rhizomorph structure, which you see in the bottom right image, which are basically threads of mycelium. Mycelium is on the photos on the left, those white sheets that are underneath the bark. That's fungal mycelium, the body of the fungus. And so those are basically encased in this black melanized rind, and armillaria uses that to spread out from an infected tree through the soil. It grows through the soil till it comes in contact with another susceptible host. And that's a pretty advanced feature for a plant pathogen. It also has the ability to produce annual mushrooms, which are edible if you eat mushrooms. I don't. So armillaria, parasite, but also lives as a saprophyte. So it has these kind of two modes of lifestyle. We have, oh, just a side note here, anytime that you have decline and recent death of a tree, because armillaria is just so abundant, here we have a white oak that recently died. It's always a good idea to just pop off the bark on the root collar or the lower trunk and just look for those mycelial fans because armillaria is so often involved in the decline and death of forest trees. Now, we don't want to give it too much credit. It's often associated with stress, and I will talk about that. Stresses that weaken trees and allow a pathogen like armillaria to finish them off. But it is just so abundant out there that it's kind of the first thing I'll usually look for when I'm looking at a dead tree, excavate the root flare and look at some of these lateral roots for the mycelial fans underneath the bark. Okay, so we have organisms that act as a parasite and a saprophyte, the way armillaria does, and armillaria is recycling a lot of dead organic matter back into the soil, so very beneficial. We also have organisms that are obligate parasites. That means they can only survive within a living host. And our rust fungi are just excellent examples of obligate parasites. What I'm showing here is pine needle rust caused by a fungus named Coleosporium, widespread throughout the Northeast, but mostly on only on 2 and 3 needle pines. Here it's on a pitch pine. And we had an outbreak a few years ago. I think it was maybe 2021, just a major outbreak. And it looks bad, but it really doesn't cause a lot of significant damage. Now, you would think organisms that have evolved to they can only survive within a living plant, they wouldn't be that successful. But rust diseases are some of the most important worldwide. They're some of the major agricultural diseases, forest diseases. So they have tremendous importance. But these organisms, they cannot live as a saprophyte. If the plant tissue is dead, they die as well. So a very interesting strategy they've adapted there to only survive within a living host. And the vast majority of the microorganisms out there including most fungi and bacteria, they are obligate saprophytes. They live on dead organic matter, and they have no ability to parasitize a living plant. And that is the vast majority. So when we think about plant pathogenic fungi and bacteria, it is really a very small subset of the overall population. Most are free living, decomposing, breaking down organic matter, and just a very small percentage have evolved to attack and kill plants. So it's important to keep that in mind as we're thinking about plant pathogens. Okay, let's tackle another term that is very important in plant pathology, and that is inoculum. Many of you are probably familiar with inoculum. A strict definition is a pathogen or its parts capable of causing infection when transferred to a favorable location. I find that kind of confusing. Basically, it's diseased plant material that harbors the pathogen and allows it to kind of sporulate and spread from that diseased tissue. What we're looking at here is a maple branch, a segment that is infected with coral spot nectria canker. Nectria cinnabarina is the pathogen. And you can see the stain in the cross section there, and we have some cracking, splitting bark. And then you see these pinkish orange pads of fungal tissue that have ruptured through the bark. So this dead branch that's harboring nectria, it represents inoculum. So we have infected tissue. It's capable of allowing the pathogen to survive and then sporulate and spread. And just for a better image here, this was on a European beach on campus. So this branch was still alive, but harboring nectria. So a very distinctive sign of the pathogen, really contrasts with the gray color of the bark. Living and dead plants serve as an inoculum source. We have here, unfortunately, a beautiful old American elm, just a few towns south of here in Amherst, that died due to Dutch elm disease, as so many have over the years. Now, whenever we have an American elm that dies from Dutch elm disease, we want to remove it immediately because it becomes a significant inoculum source for Dutch elm disease. As the tree is dying, it becomes a magnet for elm bark beetles. They infest the tree to lay eggs and create their galleries, and the fungus is present. And so when the beetles hatch and mature and then leave, they obtain these sticky spores of Ophiostoma, and then they fly off to another elm, feed in the twig crotches, and introduce the pathogen there. So this is a case where this inoculum source has to be dealt with immediately if we want to contain the disease within a certain area. Now, on a forest level, inoculum control is often not possible because we're dealing with so many infected trees in such a large area. But on an individual tree level, like I deal with, we're often focused on removal of inoculum so we can limit infections on other nearby trees. Okay, another core concept that we should talk about here is the disease triangle. And now there are three components, three factors, and they must be present for disease to occur. And I hope many of you have been exposed to this in the past. We need the pathogen. That would be, I think, pretty self-explanatory. In order for disease to occur, we need the pathogen, but we need a susceptible host, a host that is susceptible to that particular pathogen, and then we need the correct environmental conditions. And only when we have all three do we see disease develop. And that is about as basic as it gets for plant pathology. Now, it gets interesting when we look at specific examples of how this develops. So Rhizosphaera needlecast is a disease that I deal with frequently here in the diagnostic lab. It attacks a wide array of conifers, but it's most important on blue spruce, which we know is not native here in the Northeast. So the symptoms of Rhizosphaera needlecast: on the left, the purpling, browning of older needles as they're prematurely shed, and on the right, a magnified view of an infected needle. Those little clear colored masses are spore masses that are emerging from the stomata to spread. Now, Rhizosphaera thrives when we have high humidity and we have abundant rainfall that is splashing and blowing those spores from infected needles to adjacent uninfected needles so it can establish. Now, last summer, I was in Colorado. My wife's family We went on vacation there. We've been doing more family vacations since COVID, and so we went to Breckenridge, and that is where blue spruce is native to. And so we were in these high elevation conifer forests with blue spruce and lodgepole pine and bristlecone pine, and it was just incredible to see blue spruce in its native habitat. So we're at almost 10,000 feet of elevation here, so very high elevation, loads of blue spruce, as you can see in the photo here, and that's my wife for context. And I was looking at these trees over hard to look for Rhizosphaera, and they were just immaculate, just perfect, not a sniff of needle cast. And what was fascinating to me is, so I use Weather Underground to check the weather conditions. So I pulled up the app, and it never gets very hot in this area around Breckenridge. This is early August. It peaks out around 80 degrees, so never very warm there. And then what really blew me away was the dew point. So it was 76 degrees. The dew point was actually below freezing. So the air is so dry in these forests that the dew point was actually below freezing. I didn't know you could have such a wide discrepancy between the ambient air temperature and the dew point, but just very, very dry air, dry during the summer. And so Rhizosphaera is native to this area, but it causes very little injury. So we take blue spruce from these forests and we bring it to the Northeast where it's very humid, where it's very wet, and we have dramatically changed the environmental conditions. So we have the pathogen, we have the host, and now we have completely different environmental conditions, and we see disease severity just rocket upwards. And um, other conifers that are other spruce that are native to these wetter conditions are much more resistant to Rhizosphaera as a result of that. So that is just an example of how when we change the environment, we can really change the dynamics between the pathogen and the host. Okay, one thing that I really like to hammer on when I am, going through these introductory lectures is the difference between the symptoms and the signs of plant disease. The symptoms is the expression of disease, the external and internal reaction of a plant because of disease. So that's our visual cue that something is wrong. And there's a range of different symptoms that we can see on trees and shrubs, and that might be foliar wilting and dieback, chlorosis, which is yellowing of the leaves, a marginal leaf or needle scorch, leaf spots and blotches. We may see a more rare symptom as epinasty and irregular curvature of the stems, staining of the vascular tissue, like with Dutch elm disease. The water conducting xylem tissue will become stained. Branch or twig swellings, cracking, splitting on the branches, resinosis, resin flow or sap flow from twigs, branches, or the main trunk itself, or trunk and stem cankers, and we could go on. There's a range of different symptoms of disease. So that is the expression of disease by the plant. So its reaction to being diseased and infected. So we have an example here. We have an American chestnut infected by chestnut blight, Cryphonectria parasitica. So we see the cracking, splitting bark, the canker that's clearly visible. This is very obvious that it's disease. Now, that is the symptom of chestnut blight. If we look within the dotted circle here, we can also see the signs of the disease. The sign is the pathogen or its parts. So it's the actual disease agent. In many cases, the signs are not visible to us out in the field. When we're looking at a symptomatic tree, we don't see the actual pathogen, but here we can. So what we're looking at, those orange colored pads that are rupturing through the bark, very similar to what Nectria produced on that maple branch that I showed you a few slides previously. So this is a case where we see the symptoms, the cracking, splitting bark, the cankering, the swelling, and then we also see the signs of the pathogen, these pads that are rupturing through to release spores and disseminate. So I took this photo many years ago now, renting a house as a graduate student, and this tree was out front, and I saw that it was sporulating. So I cut this branch off and threw it in the compost area. And the landlord got angry because I cut a branch off their chestnut. And I said, yeah, but I was reducing inoculum for any nearby chestnuts so they wouldn't be infected. And that argument was kind of lost on them. I don't think they really appreciated that. Okay, so here we have, this is a good example of how symptoms are helpful in telling us that the plant is diseased or stressed, but they really don't point us to a particular problem. So we have the sugar maple. You can see there's scattered branch dieback. The leaves are browning. They're going to be prematurely shedding pretty soon. So we've got this kind of scattered branch dieback in this sugar maple here. This could be the result of a cankering pathogen. Cankering fungi they're after sugars in the phloem, and so they will girdle the branches, and that results in water starvation symptoms in the foliage. So browning leaves and prematurely shedding leaves can be the result of branch cankering. Oops. It could be verticillium wilt. Verticillium is a soil-borne disease that establishes in the roots and then moves through the vascular tissue and gets up into the canopy. It results in this green staining of the vascular tissue. Is very common on maples. So could be verticillium, possibly just drought. We have this open grown tree here surrounded by moisture sucking turf grass. Maybe it's just drought stress, water starvation, drought stress. That seems to make sense. Possibly armillaria attacking the root system, leading to kind of this scattered chronic canopy dieback. And so this just illustrates that there are symptoms that can be caused by a multitude of different disease agents. So some symptoms are very particular in their nature, like the green staining of the vascular tissue. That's in almost every case going to be caused by verticillium. But just this general canopy dieback could be caused by almost anything, whether that's a biotic agent, something living, or an abiotic stress. I know many of you are wanting to know. It actually was verticillium. Well, that was the cause here. Okay, let's look at Diplodia shoot and needle blight because it provides another good example of the symptoms and the signs. I would expect many of you are also familiar with this disease. It's a serious problem for 2 and 3 needle pines. It actually has a very broad host range. You can find it on eastern white pine at times when it's stressed, but I see a range of different conifers come through the diagnostic lab that can be infected by it, but usually those are pretty stressed and weakened trees. One thing really interesting about diplodia, a common symptom, is that the new growth flushes and then it's killed and dies back. So you have these needles and these shoots that are just partially elongated. So that's often a good symptom of diplodia. So when I get samples and I see that the new growth got about half elongated, a quarter, a third, half elongated and died, I'm immediately looking for diplodia because that's a typical symptom. But it doesn't confirm it outright. It's just a symptom. Here's a red pine, Pinus resinosa, that came into the lab. You can see the shoots flushed. They got about halfway there, and then they died back. So this is a symptom of the disease, an expression of the disease by the plant. Now, if we want to confirm it, that's where we have to look for the signs, and that's what I'm doing here in the diagnostic lab. And looking at the base of the needles here, you see these black pads of tissue that are rupturing forth to disseminate these small brown colored spores. So this is the sign. This is the actual pathogen itself. And when you're out in the field, when you're in your forest, you don't have, a nice laboratory with a microscope to sit down and confirm these things. So really, when we're outside, all we have in most cases are the symptoms, unless we have, something like chestnut blight where we can see the pads of tissue around the canker. But for the most part, we're just reliant on the symptoms, which makes it very hard to diagnose what's happening in a lot of cases. Diplodia can also infect the cones, which is really interesting. And so, all right, we're growing red pines for timber. If the cones are infected, big deal, who cares? What's important though is this can act as an inoculum source. So the cones fall, they're not going to break down nearly as quickly as the needles. And so this allows the pathogen to persist at the site. So it can sporulate from the cones and, the spores can splash or be blown onto nearby saplings or young trees and establish new infections. So its ability to attack the cones is an interesting adaptation that allows for persistence in the stand. Oh, oops, okay. So we love to simplify things. We do that all the time. We take these organisms, we put them into their bins, and we have these kind of preset ideas of how they operate. And one of those is that when we have a healthy host, it is able to resist disease. Okay, so we have our Diplodia inoculum, we've got infected needles and cone scales, and They're pumping out all these spores and they're close to a susceptible pine, but it's healthy. So it restricts disease development. It's able to fight it off. And that happens in a lot of cases. That is actually accurate. Um, and we also think that, okay, there's inoculum present and we have that same host, but it's stressed and weakened. It's been dry, it's been really hot, it's drought stressed, it's been attacked by insects, in this case, sawflies, red-headed pine sawflies, so it's weakened, and that makes it susceptible to attack. And that does happen in a lot of cases. But what we're learning with more and more work is that this is not always the case. And this study here is really interesting, came out a few years ago. And what it talks about, I'll just read the title, the Diplodia tip blight pathogen. They're choosing to call it Sphaeropsis. Fungal taxonomy is, it's always in flux, but I call it Diplodia, is the most common fungus in the Scots pine mycobiome. So some of you may say, what is a mycobiome? So if you think of your own body and think of the microbiome present within, there's all kinds of microorganisms inside our bodies. Well, if we were to just look at the fungi that are present, that would be the mycobiome. So they're saying that Diplodia is the most common fungus in Scots pine that they looked at, irrespective of its health status. And basically what they found is if you collect shoots and needles of Scots pine, whether they are healthy or diseased, and you grind up that tissue and you extract DNA and you just look at the DNA of the fungi that are present, you will find that Diplodia was highly abundant in healthy trees, presenting in its endophytic mode. So what that means is that you have a healthy Scots pine, and yet Diplodia is present within it. So how can that be? This is a disease-causing pathogen. We know it causes disease. It causes some significant damage. How could it be present in an otherwise healthy plant? So it really turns our thinking on its head. And the reality is, it's able to infect these trees, but then it doesn't cause disease. It just kind of hangs out and just kind of exists within the plant. And then maybe when this tree is stressed or weakened, then it kind of switches over to its pathogenic mode. So it really just kind of illustrates that there's so much we have to learn about these organisms, and the categories and the bins that we've put them in really don't fit in a lot of cases. So I just wanted to take a little side tangent there to talk about that. All right, let's get back on track now. So disease has a negative connotation. Disease is bad. We don't want disease. It's not beneficial for the forest. The reality is that a healthy forest has disease, and it's very beneficial in some ways. So what we're looking at here, a large black oak. This is out by the Quabbin Reservoir here in Western Mass, the drinking water supply for the city of Boston. We have this big black oak. It was loaded with decay caused by armillaria, and it suffered stem failure and it collapsed. And when it did that, it didn't just fall over, it took out some neighboring trees and it created a canopy gap. So now we have this gap where more sunlight is reaching the forest floor. So we will see increased regeneration of seedlings that will form saplings. We have trees on the margins of the canopy gap that have been kind of struggling for light. Now they can grow into the gap and start to express dominance. We have this kind of micro habitat that's been created with the fallen log and the stump that is good for insects and other invertebrates. And so we see this diversification in the structure of the forest. And so this is very beneficial over time. And so disease can be a very good thing in our forests. So it's often vilified. And so we have to sometimes take a step back and recognize that disease is a good thing for our forests in order for them to remain healthy. And here we've got just this ratty understory of suppressed and weakened white pines. Many of these trees, they're never going to make it up into the to the upper canopy, right? They'll always be suppressed, maybe intermediate, but they'll never reach co-dominant or dominant crown class. So they need to be removed, because what do? They just harbor disease. They allow inoculum to linger in the stand. They're never going to make their way up into the canopy. And so armillaria will take care of them. Armillaria will wipe these trees out, these trees that never have a chance to make it up into the upper canopy, and it'll recycle that material back into the soil. So again, it's beneficial side. We often view armillaria in a negative light, but in the reality, it's very beneficial for recycling and kind of taking out weak and suppressed trees from our forests. Okay, now in a lot of cases, we have created the conditions that are perfect for disease. So this little illustration on the left, you see circles of various sizes and colors. Those represent different tree species. Each color represents a different tree species, and the size represents how old they are. So the larger ones are big, mature trees, and the smaller circles are small trees. And so a lot of pathogens are fairly host specific. And so if we think about the red circles, let's call that red maple. So we have a disease of red maple, and it whacks that little small red maple. It didn't have a very good, robust defense system. It's just a small tree. So now, in order for this pathogen to spread, it's got to make its way through all these trees that are non-hosts for it. It has no ability to attack red oak or white pine or shagbark hickory. It has to find another red maple. And when it does so, it encounters a large, mature red maple, has good defenses, and the small tree that it killed was so little it didn't represent a very large inoculum source. So it doesn't have a lot of energy reserves to attack a large tree. The view on the little illustration on the right, this is a white pine plantation, or insert any other tree species, where the trees are all the same size, same age. They're planted in tight rows. So when we have a disease start to develop, if the tree is susceptible, all the other surrounding trees are susceptible. And as disease starts to develop, we have this huge buildup of inoculum. And so now we have this large population of the pathogen, and it can readily sweep through. And so our plantations, our monocultures are basically ripe for disease. And, really, there's no better example around here than Christmas tree farms. I actually really like working with Christmas tree growers, and it's given me a new appreciation for, the cost of a tree, everything that goes into it, all the weed control, the insect control, the disease control. And, they always have their meetings in the middle of the summer. And I tell you, the worst place to be on a hot summer day is out in the middle of a Christmas tree farm. It is just, there is no shade. There are bugs everywhere. It's just like, the total opposite of what you experience in the winter. So this was a tree farm. We've got this fir. We'll look at the one on the left. The one on the right is also in pretty rough shape. We see the browning shoot tips, the new growth flushed, but it didn't grow very much. It's browned out. But you see the older growth is still mostly green. I mean, it's clearly diseased, but you think, well, maybe it's got a chance to survive, but it's clearly off color. It's not as green as the trees behind it. And when we get closer, and I should have removed more of the bark because the base of it, you can see the resinosis. That's a symptom, as we talked about. And when I started removing bark, what did I find? I found armillaria. And you can see that mass on the left is a stump. So the tree that was there previously, armillaria was able to establish on the stump. A new tree went in next to it, and the inoculum was there. Armillaria was able to spread to the small sapling nearby and infect it and cause disease. And so, most Christmas tree growers, if they start from scratch, you get maybe one or two rotations, growing trees up to a saleable size, and then harvesting before armillaria starts to get in there, and it can really cause some serious problems. Okay, so stepping back for a minute, for our disease literature, where do we go to find out this kind of thing? What are the go-to resources? Number one is Diseases of Trees and Shrubs, Wayne Sinclair's book. It's getting a little older now, 2005, but it's still just loaded with information. The attention to detail is just incredible in this book, and I would definitely recommend having a copy. It's just full of great material. I actually have 4 copies. I have one in my office, one in the lab. Then, even though it's like a 15-foot walk, I have one at home, and then I have a spare copy in case any of those 3 get lost. There are more specialized books. The Compendium of Conifer Diseases. This is from the American Phytopathological Society. So that's our primary plant pathology organization. So there are specific compendia for certain plant commodities. So if you find yourself working a lot with conifers and you want to have something more in depth, this does exist there. They can be expensive because there's obviously not a huge market for this kind of literature. This is a great book here, The Manual of Woody Landscape Plants, Michael Durr. Now this, obviously a lot of ornamentals in here, but a lot of forest trees as well, just to know things about their basic biology, what kind of soil types they prefer, their growth form, the seed production, things like that. Just a lot of great information here. Michael Durr, former UMass alum. I'll also add that in there before his long career at the University of Georgia. And then, extension fact sheets. Cornell has a lot of great fact sheets. I try to keep my fact sheets up to date. That's one thing I do during the winter. So there's a lot of great university fact sheets out there. You're going to be hearing about beach leaf disease next month from Kelsey McLaughlin with New York DEC. So that's definitely something you'll want to tune in for. And then I know many of you are probably familiar with the Silvix manual. You know that its most recent update was 1990. You may not know is that it's getting a current revision. So the Silvix manual is being revised currently, and I'm actually working on one of the chapters, working on the American Elm chapter with some colleagues. And so the new chapters are just starting to trickle out now, I think later this year, but by next year there'll be a lot of new chapters there. So the Silvix manual, I'm always going to the Silvix manual to look up material, and so this is going to be significantly revised and in an online format where it can be more readily updated. So definitely check out those revised chapters as they come out. Okay, one thing we have to talk about is Mannion's decline spiral. This is a nice conceptual framework for understanding tree disease. And really, it just shows how different stresses interact, and you get this kind of cascading sequence of events that ultimately leads to tree death. Now, trees can be infected or infested by a single insect or disease and kill them, Dutch elm disease. But even with Dutch elm disease, elms that are drought stressed are more susceptible. So there's usually multiple stresses that interact to cause decline and death. And so our outer ring of the decline spiral here, we have our predisposing factors. These are usually abiotic stresses, things like poor fertility, poor soil drainage, climate change, air pollution. So these take an otherwise healthy tree and just kind of weaken it. Next, we have our inciting factors. So these are things that really initiate the decline, like spongy moth defoliation or a really serious drought or some significant levels of air pollution. So they're usually not enough to kill the tree by itself, but they really initiate the decline. And then the contributing factors, these are the things I see in the diagnostic lab. So cankering fungi, wood and bark boring insects, things like armillaria, verticillium. So these are actually killing tissue, but we have to recognize that in most cases, they are present because of the predisposing factors, because of the inciting factors. And so too often we assign, I think, a higher level of importance than is necessary to, the cankering fungi, for example. Otherwise healthy trees can usually fight them off, but when they're weakened and stressed, that's when they are most susceptible. So just a nice conceptual framework there. And a real world example from our region, certainly here in Massachusetts, is we go back to the 2016 drought, really serious summer drought that we had here in 2016. And spongy moth populations had been building in the year prior in 2015, but that drought year really suppressed the fungal and the viral pathogens of spongy moth that keep their populations low. And spongy moth just exploded. And 2017, we had almost 1 million acres defoliated here in Massachusetts. And I know it pushed west. And so parts of New York State, where some of you are, might have seen it later. I know I was camping in Vermont, in Western Vermont, 2020, and there was really serious defoliation there. And I know in Eastern New York, there was as well. So it may be a little more recent for you. But those drought stress trees that were badly defoliated by spongy moth, they were just, prime candidates for infection by armillaria. So armillaria finished off a number of those trees, especially a lot of oaks. Armillaria readily takes advantage of trees that are drought stressed and have been defoliated. Insects like the two-line chestnut borer, number 4, readily infest weakened and stressed trees, so another biotic agent there. Number 5, this was more an issue in our urban trees, but after significant defoliation events, Lacanium scales, these soft-bodied scales, their populations can really increase. So that orange arrow is pointing to this fat Lacanium scale on a red oak twig. And we had a major Lacanium scale outbreak 2019, 2020. And what is in those red circles, that's Diplodia twig canker. And Diplodia twig canker on red oak just exploded throughout this area. So from 2020 through 2022, we saw a lot of twig dieback as a result of this disease. So it all traced back to the drought in 2016. It set off this cascading sequence of events that was hugely damaging. We lost a ton of trees here in the state as a result of that drought. And there were insects and disease involved that were really hard to kind of predict at the time what they would do. And it lasted for multiple years. So you can see how these singular events just lead to these insect and disease outbreaks over time. And so whenever we're trying to diagnose what's happening, we have to also recognize that what we're seeing is just a snapshot in time. It's just that particular moment when we're there. In many cases, there are predisposing or inciting stresses that may have happened years prior or even decades prior that have kind of set the stage for what we are seeing at that time. And so without a lot of history at the site, that can be difficult to factor into what we're trying to diagnose. Okay, as I said, weather plays a big role in disease outbreaks. What we're looking at here is a graph of average annual temperature in Massachusetts. So this is the average temperature through the calendar year, January through December, from 1895 to 2025. And it's clearly getting warmer here in the state of Massachusetts. The trend is 0.3 degrees Fahrenheit per decade. So over 130 years, that's almost 4 degrees Fahrenheit. So definitely getting warmer. But if we pare this down further, if we look at the warmest average June through August, so meteorological summer, this 3-month period, look at the highest average temperature during that 3-month span since 1895. Number one is 2020, and then tied for second and third is 2022 and 2024. So it's getting warmer, but our summers are also getting warmer, significantly warmer in some cases. And that represents, in some cases, a significant stress for our trees. Warm during the course of the year and hotter during the summer. And that is a problem because they have to cool down. I am constantly amazed on a sunny day, when you're standing out in the sun and then you go under the canopy of a tree, just how drastically different the conditions are. Trees are absorbing just huge amounts of solar energy, but the leaves are only able to absorb so much and they have to dissipate heat. Otherwise, the foliage is going to overheat. And one of their primary ways in which they do that is they transpire water. So water is released from the stomata, it evaporates from the surface of the foliage, and in doing so, it transfers heat energy, lowering the leaf temperature so that the leaf doesn't overheat and disrupt all those cellular processes. During the heat of the summer, transpiration can account for more than 95% of water usage. So we think, trees have gotten through the spring period, they've got all their tissue is developed and hardened off. They shouldn't need as much water during that time, but the reality is they're churning through these huge amounts of water just to cool down during the heat of the summer. So that is potentially a problem. Thankfully, we're getting more precipitation here in the Northeast. So it's getting hotter, but it's also getting wetter. And this is average annual precipitation in Massachusetts, January through December. Again, we see tremendous year-to-year variability, but the trend is over a ½ inch per decade. So we've taken on more than 7 inches of rain since 1895. That is incredible. I mean, none of our months in Massachusetts do we average 7 inches of rain. So we're averaging now over a month's additional precipitation since 1895. So really quite a dramatic increase in precipitation. And if it's going to get hotter, we're going to need that so trees can continue to transpire and cool themselves down. But more precipitation is generally good for a lot of disease-causing organisms. So when we have excessive precipitation, that can lead to outbreaks of certain fungal pathogens and bacterial pathogens. All right, just going back to that drought for a minute. So this was a paper that came out, The Anatomy of the 2016 Drought in the Northeast. And what they discussed here, 2016, we had one of the warmest and driest summers on record. We had a historically low winter snowfall that exacerbated the drought conditions during the summer. And what's interesting when we look at climate models and we look at the data, it's getting warmer, we know that, it's getting wetter, but some of the models are also suggesting that these short-term summer droughts could increase in frequency. And these are called flash droughts. They come on very quickly, they're very intense, and then they dissipate quickly after a short period of time. But if they occur during the peak of summer, they can be very damaging. And if we look over the past, 5, 6 years, 2020, 2022, you could say that we had a flash drought. If we look at the fall of 24, where it was just historically dry, we could possibly call that a flash drought. So some of the models are suggesting, yeah, we're getting more rain, but we could also have these intense short-term droughts that could be very damaging. Okay, so I have hit you with a lot of material, and we're getting towards the end of the lecture here. So let's cool things down. We'll just look at some nice disease pictures of things that should be on your radar coming into the upcoming growing season. So springtime, new leaves are flushing. They're very susceptible to attack by anthracnose fungi. And if we have a wet spring, then anthracnose could be quite abundant. So we have a sugar maple here. The leaves are not fully hardened off. And you see these kind of dark colored spots and blotches that kind of have an angular shape to them. So again, this is just a symptom of anthracnose, but it's usually pretty distinctive. Again, here it really becomes dark in color. The angular edges to them is usually pretty distinctive of anthracnose. So keep an eye out for that as the spring season progresses. We've got a red maple here with these scattered spots and blotches. This was from a couple summers ago. The symptoms of maple anthracnose can really vary considerably, and that's because there are several different fungi that are responsible for anthracnose. So anthracnose is kind of an umbrella term where a lot of different fungi they're in that bin, as I said, the way we like to classify them. We're calling them anthracnose fungi, even though they are a little bit different from one another. So you can see certainly some variation in the symptoms here. This image on the right, we see circular spots that are coalescing to create larger necrotic blotches. So typically anthracnose on maple is not particularly serious, but if we have a severe outbreak, then in some cases we can see some significant defoliation. Now, beech anthracnose, during the early stages of beech leaf disease, we were talking a lot about distinguishing beech anthracnose from beech leaf disease, just so people were clear. And this is one of the best examples of how water moves these anthracnose spores across the surface of the leaf. And, beech has very prominent veins. And so you can see along the primary veins are where a lot of the spots and blotches are developing. So as water is passing across the leaf, it's channeling along the veins, and that's where the fungal spores are settling. And so we often see this marginal necrosis, and then we kind of see the blotches develop along the veins as it works its way back. And so good to be able to immediately distinguish this from the symptoms of beech leaf disease, very distinctive. So we can immediately rule that out. Oak anthracnose can be particularly damaging in some cases. This was a red oak that came in with distorted foliage, collapsed, blackened leaves, and also shoots. So oak anthracnose will attack the newly developing shoots before they're hardened off. So you can see the shoots on this oak were green and succulent and more easily attacked. They didn't have their bark fully developed. And so just another image here showing the blackening and the shoots with and I don't have the arrows, and the spots that are developing on those green succulent shoots. This was a white oak sample that came in where the leaves were just completely collapsed throughout most of the tree. So this was kind of an extreme example. Thankfully, oaks do have the ability to reflush. That secondary flush of growth if conditions are ideal. But having your first set of leaves completely blighted and killed is a significant stress in some cases, especially if you think about if it's been dry, if it was defoliated the previous year from spongy moth, if it's fighting off an attack from armillaria. So these things just pile up. And so anthracnose at times can be a significant contributor to decline when other stresses are present. Oak wilt, obviously a major concern to us here in the Northeast. So we want to be able to readily distinguish oak wilt symptoms from oak anthracnose. And so anthracnose is going to be more in the lower canopy where moisture and shade and humidity are more abundant, occurring earlier in the season in May and June, whereas oak wilt symptoms tend to develop from, kind of late June or July onward. So a little bit of division there and when the symptoms appear. And the oak wilt symptoms, we're going to look for water starvation from the tip of the leaf working its way back, as opposed to this blackening and collapse of the foliage that anthracnose is responsible for. Okay, some of you may not be familiar with Tobackia leaf blotch, another significant foliar pathogen of oak. I often get samples where we have Tobackia and oak anthracnose present at the same time. And so we get these kind of dark spots that will form on the foliage. And if we just take a close look here, you'll see this kind of these like black oily droplets in the center, and these are masses of spores. So the leaf spot would be the symptom of Tobackia leaf blotch, and then the spores would be the sign. That would be the sign of Tobackia. Again, it's so small that we are unable to see it in the field. We need the magnification here in the lab in order to properly see that. These spots will kind of coalesce and form some really significant blotches in some cases. This tends to be a later season disease, so the foliage is infected in the spring, but it doesn't become symptomatic until later in the growing season. But it can be a significant contributor to a leaf blight. This is an interesting example of a kind of a co-occurrence of insect and disease. So jumping oak gall on white oak, it produces these circular spots that are kind of orange brown in color. And on the underside, there's a little gall that the gall wasp larvae are developing in. And some years, white oaks will just be just tattooed with gall wasps leaf spots. And in some of those cases, you get Tobackia in addition, and it really furthers the leaf blight and the dieback. So just an example of kind of a co-occurrence and how Tobackia, a fungal pathogen, will take advantage of tissue that's been blighted by an insect. Increasingly here in the Northeast, we're seeing oak leaf blister. This tended to be more of kind of a Southeast Mid-Atlantic disease. And more I am seeing it on both landscape oak and some forest oaks. And here is the early stage symptoms of the disease, this kind of greenish, slightly reddish blistering that is developing on the leaves as they're maturing. And then as the leaves get more fully formed, you see much more distinctive symptoms. So this is something to keep an eye out for. This is a disease that seems to be encroaching up here into the Northeast in greater abundance than it was previously. In the past, if we found oak leaf blister, it was kind of a novelty, and now it's certainly becoming more abundant as a result of our changing climate. Okay, and then let's finish off here just talking about hemlock. We had an outbreak of Syrococcus tip blight in 2024 that carried over into 2025. And we see the symptoms of the disease here. The new growth is flushing, and it is killed and dies shortly after flush. So we see these pale green to brown needles, these little tufted shoots. And then we also see some shoots that are continuing to grow normally and pushing through that. So this was very widespread in 2024. And that was a result of summer weather in 2023 that was very wet. We had an extremely wet late June and July in 2023, and that really helped to promote disease development and buildup of inoculum. Now, we had a previous outbreak in 2010, and that was a result of very wet conditions in the summer of 2009. And so this is an example of sometimes we have to, the disease we're seeing in a particular year is a result of conditions the previous year that allowed for rapid buildup of the pathogen. So sometimes we have to go back to last year and ask, what were the conditions then that may have been conducive for a disease outbreak that we're seeing the following year? So another image here. This is really interesting. So there were a lot of blighted tips on this hemlock, but then as the growing season progressed, it kind of pushed through and we see normal new growth. Hemlock exhibit indeterminate growth rates. So as long as conditions are good, they will continue to put on new shoots and foliage. So as the summer progressed, it was able to push through that early disease. Isabel Monk, forest pathologist with the Forest Service in New Hampshire, did a wide survey of Syrococcus here in the Northeast and found it everywhere they looked. So it's very widespread throughout the region. It's mostly a minor disease causing some kind of minor shoot dieback. However, there were some stands, as she reported, where there was really significant defoliation of understory hemlocks. So in deep shade, where the conditions for the disease are much better. In some cases, you can see some significant Syrococcus tip blight. So something that kind of flares up every now and again after we have these very wet conditions that allow the pathogen to build up for the most part, not a serious chronic persistent disease of hemlock. And then just to wrap up here, here's another one, Fibella needle blight. This is kind of a uncommon to rare needle blight pathogen of hemlock. I'm always looking for it, but I don't often find it. The symptoms on the left, the browning needles that will hang on the shoots. So sometimes it's called a needle cast, but actually the dead needles will hang in the canopy. And if we look on the underside, these brown pads of tissue that are rupturing forth to release spores. So those are the signs of Fibella. There are reports throughout the past century where the disease flares up, but it's a good example of a native disease that doesn't cause a whole lot of issues for hemlock. Now, if it was introduced maybe into Asia on a hemlock there that has not evolved with it, maybe Fibella could cause a major widespread outbreak. But here, where it's adapted and evolved with Eastern hemlock, it just causes very minor injury. Oh, and I kind of mentioned everything I was going to say there. Sometimes you see it, needles attacked singly or across entire shoots when outbreaks develop. I've had a few photos, few samples already of some really serious winter burn on hemlock this spring, and it varies from year to year. Here's just an example of the range of symptoms you might see on hemlock, where just the needle tips are burned and blighted, like on the right, to something more significant on the left, and in the middle, just really torched branches. So be on the lookout for winter burn on eastern hemlock when it's very cold. And then my last slide here, natural senescence, which we will distinguish from the needle blight pathogens. I was looking this up. It was actually surprisingly hard to find how many years are hemlock needles held in the canopy. I really had to dig in the literature to find this. I did finally find a reference. Hemlocks will hold their needles for 3 growing seasons in full sun and about 5 in the shade. And that varies. It may be a little longer in the shade, but about 3 growing seasons in full sun, which I thought was interesting. Okay, we're at the end of our hour here, so happy to take any questions if you have them, and thanks for your attention.
Nick, that was great. That was a lot of fun. I enjoyed that. Which
Thanks.
is reminds me of when I was working on my master's degree, I went to a Northeast forest pathology meeting, and it was the first time I'd been out with a group of people, and they're just talking about how nice the disease looks. It's just kind of a antithetical to what we usually think about. It's just like, oh, here's a great example of disease.
Yes.
Um, so if y'all have questions, this is the time to start adding them in to the chat window, and I'll read through them, and Nick's going to respond while you're getting warmed up and formulating your ideas. I've got one, and then I'm going to build on one. So I'm gonna. So you said like you offered some cautions about like being careful of some of these myths and like simple statements. So I'm going to ask you to make one of those simple statements.
Okay.
So you could just like reject it if you want. One of the things that I hear from people is the concern about the potential for contagion. You mentioned, made me think of this. You were talking about the chestnut blight. I love the story about how your landlord just couldn't appreciate it when you said you were reducing inoculum load. Um, is there general guidance on that, either from just across like urban, like woodland settings, or how it seems to me that there's not the contagion is less of a concern than people want to believe it is. So maybe maybe it has to do with the disease triangle. So the next question is the interaction of disease triangle and Mannion's spiral. So maybe if you want to work those together or just take them one at a time.
Yeah, it's hard on a forest level to to deal with this in some cases. And so early on with beech leaf disease, people were asking, should we remove beech, right now, especially understory trees, to try to reduce some of the potential inoculum. And it's so widespread, and there's just so many nematodes that it would really have kind of little effect. And there was an interesting case of, when hemlock woolly adelgid was working its way up into central Massachusetts. There was a lot of discussion at the time of, pre-salvage harvesting of hemlock to try to get it out of there before it all died from adelgid. And what happened was the adelgid kind of stopped in central Mass. It didn't move northward as quickly as was expected. And so in some cases, these preemptive strategies really don't bear out. But on a forest level, it's really tough because you're just dealing with so much inoculum. Um, there are examples like, when boxwood blight first developed, there were recommendations about, bagging up infected plants or burning them or burying them. And there have been, widespread studies about when you have root disease in a stand, like ripping the stumps out and trying to reduce inoculum that way. The results are really mixed. So trying to now something like with Dutch elm, the feeling, if you can chip the wood down to a small size, you're going to kill the bark beetles and you're going to get enough mixing of microorganisms where you're going to kill the fungus. So there are some cases on a tree level where you can reduce inoculum and potential contagion, but on a forest level, I think it's just way too difficult. Especially when we think about Mannion's decline spiral and all the interacting stresses that are present there. And so I don't know if I answered the second part of your question very well.
Um, well, so it struck me, well, before we like, so you just like, I got to do a follow-up on the first one. Um, one of the things that people in forestry communities are talking about in an effort to try to diversify and to ensure regeneration is what we're calling different things like enrichment planting. So you go in after a harvest and you plant trees.
Mhm.
How much concern should we have for the presence of armillaria? And you showed the great
Oh,
example of the Christmas tree plantation.
yep.
Do we need to be, is there somewhere in my mind, there's something like if you wait a year, you let, the armillaria becomes less whatever virulent, or do we need to stay a certain distance away from stumps,
Yeah.
or do we need to not worry about it at all, or just or that's a lost cause, so don't even bother planting into a harvested forest?
Yeah, those are great questions. And, armillaria is just so abundant that it's like impossible to avoid. And, so many healthy trees have been challenged by armillaria and are able to compartmentalize it and ward it off. So I think if I was managing a woodlot, I would recognize it's there, but I would just, I would move forward with my replanting or regeneration efforts and just say, some are probably going to get knocked off, but if we have good conditions and the stock is healthy, that many will be able to survive and push forward. And going back to the first part, diversity is a disease management strategy. If you have a diverse forest, it's going to be less susceptible to a major disease outbreak because a lot of diseases are relatively host specific. Now, something like armillaria is has a huge host range, and then something like spongy moth has a huge host range. So there, some of our most significant forest pests and forest pathogens they have a broad host range. So even if you did have a diverse assemblage of trees, it could still kind of tear through them. So um, you know that it's really challenging to try to focus regeneration or replanting efforts around a particular disease. I think in a lot of cases, just have to kind of move forward as best you can.
All right, that's good to know. So, all right, the first question, Mandy, and I think this came up when you were talking about the maybe about the chestnut, but just what's the best way to get rid of inoculated wood? So as you'd mentioned composting,
Yeah.
does it vary by the pathogen, or what's the best way to do that if you're trying to reduce the load?
Yeah, usually infected foliage, composting, just covering it, physically covering it, just promoting decomposition. It'll break down. Once other microorganisms get in there, they'll compete with the pathogen. So that will reduce it with, more durable material like branches and the trunk, usually chipping them up or burying them or burning them if it's a significant concern. But usually physically covering it will just kind of suppress the spores spore masses from developing and dispersing. But usually when they're just left in place, that's when they're able to sporulate and spread and represent the highest risk.
Okay. Next question, concerning the diseases in trees. Is this a natural occurrence, or are these caused by human interactions within the natural environment?
Yeah, there's a lot of natural disease out there. A lot of things are just outside of our control. Unfortunately, there are a number of diseases that we have introduced. Um, And then also the plantation settings, we've created these environments that are not natural, that wouldn't occur if we weren't here. And so that helps to promote disease. So yeah, usually our most serious diseases are ones that are introduced, like Dutch elm, or chestnut blight or beech leaf disease that we're dealing with now, where the trees just haven't evolved at all. They have no resistance. So for the most part, it's the introduced diseases and not necessarily what we're doing on our own.
Okay. Um, so Lou Ward had a question about armillary, but I think we've probably covered that. Lou, you can ask if you want to re-ask it in a different way, you can. And he had brought it up again, so I'll pass that one. Daniel wants to know when you're disinfecting tools between work on different individual trees, what do you recommend? So alcohol, bleach, Lysol, some combination, something else. Any guidance?
Yeah, here in the lab we use ethanol, but you can't readily obtain that. So isopropyl alcohol, I think, is would be my preferred choice because it will readily evaporate. It's very effective. And then bleach can be very effective, but it will rust the metal. And so you have to be a little more concerned with that. And certainly, the exposure to yourself afterwards. But isopropyl alcohol, and you don't want it to be 100% because it will evaporate too quickly. You want to dilute it with a little bit of water. So generally, we keep 95% alcohol or 70% in some cases, so the water will allow it to persist on the surface a little longer. So alcohol, but bleach does work quite well. You just have to rinse it off afterwards. But just putting tools out in the sunlight, UV light will UV irradiate pathogens that are on there, and just soap and water in some cases is enough in many situations if you want to sterilize your tools. But I think if you're looking for something to zap bacteria and fungi, that alcohol is the way to go.
Is there residence time or just a contact is sufficient, you know?
Yeah, usually contact and then several seconds on the material. So if you can let it sit for 30 seconds or a minute or even longer in some cases, and you'll see it start to evaporate on its own, but that's where the water will help it persist there. Sometimes if it's on there just too quickly and evaporates, then it didn't fully do its job. We'll also flame sterilize things at times, but in general, it's not practical for the field to be doing that.
Okay. What's the role of early diagnosis and breaking the disease cycle? I mean, like, are there strategies for that? I mean, it seems and you made a really good clarification between symptoms and signs. So we're going to see the symptoms first. By the time we see the symptoms in any magnitude, it seems like the disease has progressed. So where does early diagnosis fit in?
Yeah, that's where, so I'm mostly working on individual trees in urban and suburban cases with an arborist. So when we see early symptoms and can make a diagnosis, they might step in right away and spray a fungicide or do something to chemically combat that. And this brings up a good point that I wanted to touch on. So thank you for the person who asked this, is that when you're working with conifers, it's really challenging because often the symptoms appear long after the damage has been done. So that Christmas tree that I showed that still had the older growth was still green. It was off color, but it was still green. The base of that tree was just like a sponge. I mean, it was dead, but it still looked like it was alive. And so conifers are tough that way because the needles are naturally water, they're retaining water. So they often show symptoms, well after the damage has been done. Whereas deciduous hardwoods, they're going to start wilting or showing symptoms much sooner. So there are certain cases where we can intervene, but it's usually on a tree by tree level. And so on the forest, on a forest stand level, if you started to see like an anthracnose outbreak, there's not a whole lot you could do at that time. You could think for the future, and I'm sure Rob is going to talk about this with the white pine. With white pine needle blight, there's a strong correlation between you could reduce disease severity by increasing light. So you thin out the stand, more light, more air, and wind exposure on the white pines, and needle blight incidence and severity will go down. So usually it's more of these long-term management objectives we have with forest trees, and usually not a whole lot we can do in the short term.
Is that the case for, I don't know why, like, armillaria scares me. So if you go in, you see some early signs of armillaria on a tree,
Yeah.
take a little, a pocket knife or a hatchet or something to make a section out of the base. If you've got armor there's not much you can do at that point, is there?
There really isn't. No. I
Okay.
mean, you can try to improve
Start.
the vigor, boost the vigor of the tree. And that's where with single tree management, we can water it, we can fertilize, we can try to excavate the root flare and do some other things on a forest level. You can fertilize on a forest level, but you're not going to be out there watering trees. And so, yeah, armillaria is usually a slow, chronic death sentence, and a lot of trees are able to fight off armillaria. The vast majority
Right.
of the trees in the forest are challenged in some way and able to contain it. But once they're beaten down by drought and defoliating insects, they're served up.
Right. And you made a great point about the kind of the ecological place that armillaria plays in,
Mhm.
Paul Mannion used to say it, a healthy forest has a good amount of disease in it. So,
Absolutely.
yeah.
Yeah, and what's really funny, you mentioned the Northeast Forest Pathology Working Group. Sadly, is no longer meeting, but we did a long time ago. We had a meeting, and it was at Paul Mannion's Christmas tree farm, and
Oh,
he didn't do a single thing to control disease there. I mean, the grass was up to your chest. The trees were just badly diseased, and he just kind of left it that way to see like what they would do, and the ones that were decent would sell, and most were just looked awful. So it was really kind of funny.
that's classic Paul Mannion. All right. So, John, you're talking about diplodia in this was when you're talking about the study out of Germany and the kind of the prevalence of diplodia. Um, I'll just read it. Fungi abundant in healthy trees such as diplodia, is this waiting in a dormant state, or is it coexisting through other means, such as an unknown alternate microbial host to survive? And in the dormant state, is it reproducing asexually? Is it able to spread when it's in this low energy state?
Oh, that's really interesting. I don't know if they're able to. They must be able to spread, and population is increasing in some ways if it's the most abundant. These endophytes are just this kind of weird world that we don't fully understand, where these fungi, these bacteria that are living within plants, they're not. They're certainly benefiting by having a host, but they're not. There's no mutualism. They're not providing any benefit for the plant, but they're just kind of hanging around and waiting. They're just kind of, they are dormant. They're just kind of dormant and just kind of, the feeling is they're just kind of waiting for the plant to become stressed. And there's a signal that triggers their kind of switch over to become pathogenic and virulent. But in terms of how they are spreading or reproducing inside, I don't know. That's a good question. But to be the most abundant fungus within a healthy plant like that, you would assume that it's reproducing or spreading in some capacity.
Right, right. So here's another armillaria question from Amanda. She wonders if you have any opinions on whether there's a relationship between the prevalence of armillaria and the suppression of fire in our northeastern forests.
Yeah, definitely, definitely. When you look out these pitch pine forests in eastern Mass, out on the Cape and the South Shore, they're just choked with all these little pitch pines, and now southern pine beetle is establishing and starting to wreak havoc, and they're wanting to reintroduce fire. And to see the thinning in the fire, like really what it, you take it down to, the basal areas are so low. I mean, historically, there would have been just so few mature trees and then the scrub oak underneath. So yeah, fire suppression and this proliferation of understory development that's not getting killed back by fire, I think has led to, in some cases, a lot more armillaria, but just in general, a lot of other disease and insect pests. So definitely fire that can help clear out some of these weak and suppressed trees would limit what armillaria has and kind of keep it tamped down to a certain extent.
Okay. Lou wants to know if there are pathovars. You'll have to define that of diplodia.
Yeah, so in some cases, with certain pathogens, we have this Linnaean taxonomic system that ends at species, but sometimes there are subspecies or these subgroups or races or varieties or pathobars where there's further subdivision downward. And there has been some interesting work on Diplodia, these individual genotypes, so these individual genetic units, um, where once they attack like a non-pine host, say they're on like a spruce or something like that, they tend to exhibit a higher level of aggressiveness, and then they can spread back to pine and they kind of behave more aggressively. So there is, with Diplodia, there's quite a bit of genetic variation within an individual species, and so you do see behavior vary at times depending on your location.
Okay. Um, John's wondering, this is maybe a little outside of what you look at, more of a precipitation question. You talked about increases, overall general increases in moisture, but he's wondering if that's coming as more flashy events, so like sporadic kinds of um, rain events versus kind of a more uniform distribution. Is that, are you any?
It certainly could be. So yeah, I was just showing the average amounts. And so yeah, one thing that we're also dealing with is just the incidence of intense precipitation events. So events that where we're getting more than an inch per storm. So those have gone way up. They used to be far less frequent than they are now. And so overall, it's more precipitation, but also the frequency of these really intense rainfall events has been going up as well.
Okay. Scott's wondered if you can describe any kind of a interaction between timber harvesting and pathology. Is there
Yeah,
benefits?
so
Was it beneficial occasions, detrimental occasions? Just maybe some just, I mean, that's a big subject. So,
yeah, in some cases it helps with disease, like the example with white pine, when you thin them out and reduce the number of trees per acre where they have more light, more space, you see a reduction in needle blight diseases. So in some cases you create a healthier stand when by harvesting, by removing trees. Um, in a case, you could make an argument, in a case with armillaria, and it's more, there's better examples out west with some of the root diseases out there, after they harvest these conifer stands, and mostly it's with clear cutting, but you just have this tremendous buildup of these root disease pathogens on the stumps, and then they replant, and the resulting cohort just gets kind of wiped out. So there are situations where the harvesting more intensive harvesting does create a more serious disease issue, but kind of patch cutting and selective harvesting, I think overall would reduce disease and make the trees more resistant to attack by giving them more space to grow and develop.
okay. Going back to an earlier question about how to take care of diseased plant parts. If you burn them, so if you prune them and then burn them, is there any risk of the disease spreading in the air?
Generally, no. No, once the fire hits it, it's going to kill the pathogen, and there would be a very, very low risk from burning.
Right. All right. Lots of people saying great presentation.
Right. Glad it was worth your time.
Yeah. So here's a question about continuing ed credits. So I send documentation to the people that requested it for continuing ed, and for the SAF members, I submit to SAF. For anything else, the participant will take the documentation and submit to that to your credentialing body. Arna wants to know how important are hemlock diseases for hemlock mortality compared to hemlock woolly adelgid versus the hemlock scale. So disease versus insect.
Yeah, hemlock has relatively few, if any, really serious diseases, and the adelgid and the scale are by far way more important. So they'll obviously severely weaken, kill trees. The diseases of hemlock are usually kind of a trivial matter. At times, if you have a tree badly weakened by drought and scale and adelgid infestation, and then you get tip blight and needle blight, that could be a more serious issue. But even hemlock's very resistant to attack by armillaria. It puts a lot of resources to defense against disease. And that's why, these introduced insects is just it's just a gut punch in some ways because they do naturally such a good job of resisting insects and disease, but they can't keep up with these non-natives.
All right. Sammy has a real life example, a pitch pine site that's been hit with diplodia.
Hmm.
The arborist instructed Sammy to treat with a fungicide as the
Hmm.
spores are in the cast needles. The needles have been cast onto the ground.
Yep.
However, it sounds like diplodia won't inoculate from dead needles. So, like, any, did you
Yeah, they
follow that?
definitely, the fallen needles, if they're harboring the pathogen, it can produce those black pads and disseminate spores. They'll break down more quickly. So as opposed to the cones or material that's hanging up in the canopy. But, needles on the ground can certainly represent an inoculum source for that next growing season. Um, yeah, it's interesting that fungicide application on a forest scale, and maybe these younger trees, or it's interesting to hear that that was the recommendation.
Okay. Lots of people saying great presentation. Um, so there, so here's a specific question about how to submit samples to Cornell, which I'm not going to ask you to answer, but just so if somebody's in Massachusetts, I'm not encouraging New Yorkers to do this to
Yeah.
inundate your lab. And you may have a like a zip code sorting that just deflects all those samples away. If somebody's in Massachusetts, how do they submit a sample to you?
We have a website, Plant Diagnostic Lab, where there's instructions online. There's a submission form and a link for payment. It is a fee-based service, as is all of the diagnostic labs, save for a few across the country. But yeah, all the information is online in terms of what to submit. And, generally more material is better. The more material you can submit, the easier it is to figure out what's happening. And increasingly, people are sending me photos along with the sample, which is really nice to get some context because, open a box with a bag of, dead branches and needles and try to piece it all. But the case history is really important. So, all the possible stresses that may have interacted with the possible disease to create what we're seeing at that particular time is important. So I try to get as much case history out of people as possible.
Okay. And Cornell has something similar. So I think it's called a wet, like, so just they can do a search for, if you're in New York, Cornell Plant Diagnostic Lab or
Absolutely.
something like that.
Yep.
So,
Yep. The Cornell lab. Yeah, they tackle a lot of disease
right.
problems, so they're very well suited for that.
Yeah. So, um, so Kaylee says, great presentation. There's a cherry birch in the Niagara region in Ontario, Canada, that leaves out perfectly. But as the season progresses, the leaves curl and dry up. We actually have like an ornamental apple that does the same thing. The crown is green and fine, and the tree's been growing well for 10 years. There's no dieback. So, like, what's going on?
Yeah, some trees can be diseased every year and, ultimately be largely unaffected. So, especially for the later season diseases when growth is mostly complete for the year and they've generated their starch reserves, if they develop some foliar disease, it may have some impact on growth, but it tends to be fairly minimum. And we can tolerate a lot of disease out there, and we try to talk about that through the diagnostic lab. We don't need to hose down everything with fungicides just to keep them in a perfect state. Some disease can be tolerable. We just want to keep an eye on it if it's potentially something serious. But then, some years things just explode, and you'll have years where maple anthracnose is not a big deal, and then the conditions just align perfectly, and it explodes, and it's a huge problem, and then next year it's back to being kind of its usual self of not a big deal. So it can be really hard to predict when things will break out. But if the tree has some disease or some issue, but is otherwise healthy, then may not require intervention.
All right. Joan is asking about, and I've not heard of this, maybe you have artillery fungus.
Yes.
So
Yep.
information on the control of artillery fungus.
Yes, many of you are probably familiar with artillery fungi, even if you've never heard of them. So if you have light-colored siding on your home or a light-colored vehicle, and you see these tiny black specks that are stuck to them, that are, you can scrape them off, but it takes some effort. Those are artillery fungi. So they're these little cup-shaped um, fungi that are in usually fresh mulch, and they're really interesting. So they will actually point their spore-bearing structure kind of towards reflected light, and they project their spore mass out, and it's this little sticky mass, and it gets stuck to the siding and stuck to vehicles or anything else that's nearby. So um, they're really, really difficult to manage, and people have spent a lot of money and time and effort trying to, scrub them off siding or power washing, and, fungicides really aren't helpful. It's really the fresh mulch. So if you can disturb the mulch and keep them from forming, and they're also known as bird's nest fungi. If you look down in the mulch and you'll see these little, it looks like a little curve, like a little nest-like structure with these little kind of rounded cups in them. Um, and they kind of open up and then they shoot these spore masses out. So they're actually really interesting, but they can be incredibly annoying. So usually just disturbing the mulch will be enough to disrupt them, but scraping them off siding and vehicles is a real task when they're abundant.
Okay. Neat. Amber wants to know, do you still have time for more questions?
Sure. Yep, I have a little
Oh,
bit more time
okay.
here.
Okay. Um, if somebody wants to bring saplings onto their property, probably like in an urban setting, like what are the kinds of questions they should be asking the nursery to limit the potential spread of the pathogens?
Yeah, that's tough. So yeah, usually nursery stock, I give it a hard look. And, there's a lot of diseases that spread through the nursery trade, and it's not because they want it that way. It's because, just propagating that many plants in such tight quarters creates a lot of conditions for disease. That's where, a pathogen like Phytophthora it's really been a challenge where it can develop at woody plant nurseries. There's a lot of big nurseries out in the Pacific Northwest. They are shipping material all over the country. And so Phytophthora morum, a very destructive root disease pathogen, a cankering pathogen, can be and has been spread that way. But thankfully, it's still restricted to the Pacific Northwest. But that's where just really scouring nursery stock. Sometimes if you want to bring it home and leave it in the pot for weeks or even a few months to kind of see if any symptoms develop, that is one strategy. But unfortunately, a lot of cases, a lot of disease is spread with nursery stock, and it's kind of unavoidable in a lot of situations.
Okay. Um, so three final questions, kind of on the more detail than like I wouldn't even know how to start with these. So if you could, Daniel wants to know if you could talk about the lethality of hypoxylin on oak trees in the Northeast. He says the literature describes it as quickly precipitating mortality, but they see it on declining branches without rapid spread. So
Yeah, that's
hypoxylin
not
of oak.
hypoxylone canker of oak. That's not one that I have spent a lot of time on. And I would, yeah, I would kind of agree with what your observations that it's kind of a secondary cankering pathogen that is not particularly serious on a lot of trees. So there's a lot of cankering fungi that will kind of work away on older, stressed, and weakened trees. So hypoxylon on oak to me is not a serious pathogen. I'm sure there's cases where it is, but for the most part, I see it as kind of a secondary issue that's not a major issue for oak health in our region.
Okay. And this may be a follow-up to that. Ian says his understanding is that I'm not going to try and pronounce that hypoxylin. It's biscognioxia
Mhm.
slash
Sure.
hypoxin canker of oak is similar to diplodia, that it can lie dormant. He's wondering if this pathogen is prevalent enough that removing the inoculum isn't really worth it. So is reducing stress on the host the only effective treatment?
Yeah, reducing host stress will help in for so many different diseases. And as I said, there's so much that remains unknown about a lot of these fungi. It's likely that there's a whole slew of other fungi that can live dormant within a healthy plant as an endophyte without causing disease. And we just don't know that because it hasn't been carefully studied. It's probably far more likely than we know. Um, so that could be the case. But keeping trees healthy, I mean, it sounds like a no-brainer. You keep them healthy, you keep their vigor high, and they'll be able to ward off disease on their own. But it really is a disease management strategy to focus specifically on the tree health instead of necessarily combating the individual pathogen, allowing the tree to use its natural defenses to compartmentalize or restrict development as opposed to targeting the pathogen itself. But removing inoculum, if it's possible, is always a good idea because you're just eliminating a potential source of infection that can spread within that tree or to other nearby trees. So I'm usually a proponent, a fan of inoculum reduction.
Okay. Last question. What causes winter burn?
Is a good question. So winter injury can be caused either by just straight up freeze because it's so cold. So we have just really extreme cold temperatures that cause freeze damage within the cells. And we certainly had some very cold temperatures this year. In other cases, we see wind desiccation as primarily responsible for it's just so windy that the foliage, especially needled evergreens, just gets so dry and desiccated that it causes a burn. In other cases, we see a midwinter thaw where it warms up, and especially on the south-facing side, it warms up and the needles warm, say on hemlock, and they warm up to a point where they need to transpire to cool off. But if they lose any moisture, they can't replace it because the roots and the soil are frozen. So they can't draw up any new water. So in some cases, we see it. It's usually a combination of just straight up cold temperature, ambient air temperatures causing freeze, the desiccation from the wind, or a midwinter thaw that will warm the foliage or help to break some of the winter hardiness. If it warms up, trees start to deacclimate, and then if it gets cold again, then they suffer that injury. So thankfully, this winter it was consistently cold throughout. We didn't have that midwinter warm up, and so when we look around at some plants that we normally see winter burn on, even though it was very cold, they appear to have weathered the winter very well because they just remained in that hardiness state for the entire winter.
Nick, thank you so much. This has been fabulous. It's the depth of the questions, I think, illustrates how interesting this topic is to a lot of people, and we appreciate your um, endurance
Thanks.
with the questions. We topped
Absolutely.
out. I saw we topped out at about 188 people. So that's
Okay, that's great.
a great impact. And I don't know if I told you, I send you a really detailed report after this
Okay.
is all over. So
Well, very good.
thank you.
Thank
And we'll see Nick back at 7 o'clock tonight if anybody wants to see the presentation again. Um, welcome back. So thank you again, Nick. See you in a few hours.
All right. Thank you, everyone. Bye now.